Hi everyone, and welcome to my very first video on biological processes. In this video, we'll be going over the events that lead to the invasion of the endometrium by the developing embryo. More specifically, we'll be looking at the working mechanism of the urokinase plasminogen activator, which is a short-chain protease secreted by cytotrophoblast cells. We'll call it UPA. To start us off, let's quickly remind ourselves of the events that precede this invasion. It's important to note that there are very important stages that can be seen in this photo, but we're not going to talk about all of them as they are not important for this video. Around here we have fertilization, and right after that the first cell division occurs. Over the next couple of days to weeks, we get further proliferation and eventual differentiation into a blastocyst. This blastocyst eventually reaches the uterus, where it will interact and implant itself on the uterine lining. Um, this, guys, is really where all the fun happens. So let's zoom in right here and look more at some structures of the blastocyst. It's important to note that we're not going to talk about all the structures here, but we'll talk about some that I think are important for this video. So here we have the trophoblast. This trophoblast will eventually differentiate into a cytotrophoblast that we'll talk about later. We also have the epiblast that will develop into the embryo. And here are the cytotrophoblast cells that are interacting with the endometrium. So here we'll look closer at this interaction between the embryo and the endometrium. And since we're talking about ECM degradation, or more specifically ECM remodeling, let's look at the matrix and some of its associated proteins. Some of these proteins, just to list a few of them, are fibronectin, laminin, uh, vitronectin, collagen fibers, proteoglycans, fibrins, and many other proteins. And all of these, again, will need to be degraded for successful invasion of the endometrium. So let's take a closer look at this invasion process and all of the, the proteins that are involved. This is where things get a little more complicated, so do not blink. Here we have a cytotrophoblast cell. Of course, there are many of these, but we'll just focus in on this one cell. Starting off, the cell receives signals from its environment. These can be paracrine factors, growth factors, and other hormones. These hormones will be amplified starting at the cell membrane, most likely by a G-coupled receptor that will eventually reach the nucleus and induce transcription of UPA, which is the urokinase plasminogen activator, UPAR, the urokinase plasminogen activator receptor. Now this receptor will be translated and anchored to the cell membrane, and UPA will also be translated and secreted into the extracellular matrix. At this point, UPA is actually inactive and needs to be activated before it can interact with its receptor. So UPA will be activated by plasmin. Now, plasmin at this point is at a very low concentration in the extracellular matrix, and we'll see why in a little bit. So here, plasmin activates UPA, allowing UPA to interact with its receptor, forming another protein complex, and it will be this protein complex that will actually cleave the ubiquitous plasminogen into plasmin. Now it will be this plasmin, which is now at a higher concentration in the extracellular matrix, that will do uh, three things. It will first degrade the ECM. It will also bind and activate other proteins that will help in the ECM degradation process. And as noted earlier, plasmin also acts in a feedback mechanism to activate UPA into its active form. This is all happening in a time-dependent manner, and downstream from this, we have mom cells. Where would we be without mom, right? So here, mom cells will be kind of counteracting the invading blastocyst. So mom cells will secrete PAI 1 and 2. PAI plasminogen activated inhibitor is very important because we don't want the blastocyst to invade the endometrium further and further. So PAI will bind UPA and pretty much stop plasmin, from binding UPA and activating it. So at this point, ECM degradation will stop, and the blastocyst will continue to develop.